Então, bom, pessoal, boa tarde a todos, então. Este será, então, o nosso último seminário do semestre, né? Eu vou falar em português, fazer a apresentação, então, da doutora Virgínia Miguel. A professora Virgínia, ela é investigadora adjunta do Instituto de Investigações Biológicas e Tecnológicas do CONICET, Laboratório de Bioinformática e Sistemas Complexos. É professora assistente da cátedra, seria disciplina de Química Biológica, Faculdade de Ciências Exatas, Físicas e Naturais da Universidad Nacional de Córdoba, na Argentina. Ela é doutora em Ciências Químicas, é bióloga pela Faculdade de Ciências Exatas, Físicas e Naturais, por la Universidad Nacional de Córdoba também. Sua linha de investigação é caracterizar, através de ferramentas computacionais, os mecanismos moleculares que regem a seletividade de inseticidas, seja em nível específico, tendo em conta o reconhecimento por um sítio determinado do receptor, como também em nível inespecífico, considerando a diferente composição das membranas de células de mamíferos e de insetos, nos quais se encontra o receptor. Se pretende, assim, avançar no conhecimento dos mecanismos dos inseticidas GABAérgicos, muitos dos quais amplamente comercializados sobre os receptores GABA-A de insetos e mamíferos. Creio que foi todo, doutora Virginia.
Yes.
Y bueno, le paso la palabra para su charla en el día de hoy.
Perfecto. Bueno, gracias. Buenas tardes. I will be talking in English, and I hope that's easier for you than my Spanish. If there are some parts that I'm speaking not clearly, please interrupt me and ask me if there's something that is not clear. Well, as Eduardo was saying, we work with GABA receptors. Today, I'm going to tell you a little bit about some of the strategies that we use in our lab for the screening and for the characterization of selective GABAergic insecticides. We, as mentioned, we are a biophysics group that has a bioinformatic area, but we mix experimental biophysics with computational biophysics. Today I'm going to show you results of the thesis of Salome, that she works in the lab, and some results of Marcos, who has worked, who has done this research to get his biologist degree. And in the group also there are some experimental biophysics like Daniel Garcia and Marilu Sanchez, and this is all the rest of our lab, and this is the guys that do the bioinformatics. Okay, so because of the rise of permanent resistant strain of insects, there's a huge demand of the discovery and the design of insecticides that are highly selective and that present low toxicity to mammals. And therefore, we focus on some in the discovery of new insecticides that can control vector insects such as Aedes and Triatoma that transmit neglected disease that are locally important in Argentina and in all South America, like dengue and Chagas. And most insecticides target the insect nervous system. Therefore, we work with GABA receptors that's of our interest. A GABA receptor, or gamma aminobutyric acid receptor, is the main inhibitory receptor of the central nervous system of vertebrates and invertebrates. And it's a pentameric channel, a pentameric chloride channel that opens upon gamma-aminobutyric acid binding and allows hyperpolarization of the neuron. It's a member of the cis-loop family, and it has different pharmacological states that are open, closed, desensitized. And this channel is composed in vertebrates of 19 different subunits, while in invertebrates it's mainly composed of a single subunit that is called RDL, and those difference in vertebrates and invertebrates allow the design of selective insecticides. And these insecticides, they act as non-competitive antagonists because they bound to the receptor and inhibit chloride to enter to the cell. Therefore, they provoke the insect to convulse and ultimately to die. So we are interested in knowing how these insecticides work and to be able to discover new insecticides. But since we're talking about transmembrane protein, we can not only consider the ligand receptor interactions that arise upon these insecticides binding to the receptor, but since the receptor is a transmembrane protein, we also have to take into account some non-specific regulations that can arise because of the ligands partition into the bilayer. Because as we will see next, some of the binding sites of the receptor are embedded in the bilayer, and therefore they have to access to the bilayer before they can bind to its pocket. And also, we will see that there are different compositions and different phase behavior in different lipid bilayers, and these differences can affect not only the access of these drugs to its binding site, but also some conformational changes can arise upon different lateral pressures in the bilayer. Today I'm going to show you some of the strategies we have used to study a family of GABAergic insecticides that are called isoxazolines. Isoxazolines are a recently new insecticide family that presents high selectivity, and it has become a very useful scaffold because of this selectivity and its strong insecticide activity. Isoxazolines bind to the transmembrane region of the receptor between 3 different transmembranes, alpha helices, and this is we have here marked the binding site in the lateral view and in a superior view of the site. We were interested in knowing how these insecticides bind to its pocket and which are the characteristics interactions that allow this family to be highly selective. And we use molecular dynamic simulations of different insecticides of this family, such as fluoralanar, afoxolanar, lotilanar, and sarolanar. And we use docking assays to to obtain GABA receptors bound to these insecticides. The receptor we used was the GABA receptors of Aedes aegyptis, and we use an homology model obtained since there are not available crystal structures of this receptor. We model this receptor, and we use docking assays to obtain the complexes of the insecticides with the receptor and perform molecular dynamic simulations of the receptor embedded in POPCV layers that are the main constitutives of bilayers. And we analyze the interactions that were occurring between the insecticides and the receptor. We analyzed along the molecular dynamics, which were the interactions that were generated between the 4 insecticides and the protein. And also we analyzed how stable the interaction was for each of them, analyzing the root mean square deviation of the position of the insecticides in its pocket binding site. And we were able to see that the insecticides were, here we show you only one of the insecticides, but the 4 of them were very similar, and they remain in the pocket where they were bound in the docking assays. This was indicated that interactions were reproducible in the receptor. And we also analyzed the residues, the amino acids that were interacting with the isoxazolines in the GABA receptor. Since it has been reported that hydrogen bonds are highly relevant for the interaction of isoxazolines, we made a particular analysis of such interactions, and we determined them. And also we made calculations of the free energy of binding of all the 4 isoxazolines that we studied. And therefore, this allowed us to characterize the interactions that were occurring in the receptor with the insecticides, and also it gave us an energy value that we could use as a reference for future compounds that we'll analyze to decide if new compounds have the energy of binding similar to the ones of isoxazolines that have insecticides activity. Okay, now up to this point, we have been characterizing compounds that we know are insecticides, right? We are validating the molecular model that we are using, the bioinformatic molecular model that we are using to validate that we can say, okay, if we do MD simulations with this receptor, we can say if an insecticide binds to this site or not, because we can characterize the interactions and we can determine the binding of free energy. Also, we saw that this binding energy was correlated with the potency that these insecticides present in their in vivo activity. Fluorananar is the most potent insecticide, and we found that it was the one that in our model had the highest free energy binding, and its potency as insecticide is followed by sarolanar and afoxolanar, and ultimately lotinanar. So it was reproducing the insecticides in vivo activity. Okay, so we validate our model. We say we can see which residues are responsible for these insecticides binding to a GABA receptor of Aedes. We can calculate their energies, and the model seems to work to reproduce what is observed in vivo. Okay, now, but what we really want to do is to search for new insecticides. Okay, so how do we search for new insecticides? We use a technique that is virtual screenings, right? That it uses molecular docking to search in a large library of compounds for those compounds that have a high affinity for a particular binding site. So this in silico strategy allows to speed up the process of drug discovery. So we use our model, our molecular model of GABA receptor of Aedes, and we perform this virtual screening to say, to look for new compounds that could potentially have an insecticide activity. Okay, and now for this virtual screening, we use a protocol that was hybrid, because there are different types of virtual screenings. Some virtual screenings are, as I said before, we use the receptor and we search for ligands that can bind to a particular binding site. But this is called a structure-based virtual screening because we use the protein that is the target of those ligands. But for some statistical reasons, for this kind of virtual screening, a large quantity of ligands that are known to bind the pocket is needed. So as isoxaline family is recently new, the ligands that have experimental energies determined that can be used as a test to say, well, okay, this virtual screening is working. It's very low, so we couldn't make this directly. So we use an hybrid protocol where we use a ligand-based virtual screening, followed by this effectively this structure-based virtual screening. And what this protocol consisted in the design of of a pharmacophoric model. Here we have in the first stage we use the sorry, I don't know what happened with the presentation. I'm not seeing the oh, sorry. Okay, here. Okay, so what we did is for the ligand-based virtual screening, what we did was we took several isoxalines that are known to have insecticidal activity, and design a pharmacophore that has the main characteristics of these compounds that gives them their biological activity. And we validated this pharmacophoric model using this pharmacophore to for an essay where we tested if this pharmacophore was capable of differentiating in a set of several compounds those that were we knew that were active from those that are called decoys that we know that are inactive. And the pharmacophore work, it could differentiate those compounds that were active from those that are inactive. And once we validated the pharmacophore, we used the pharmacophore as a filter to search in Zinc library that has a large quantity of compounds. We selected compounds that were purchable, so we could acquire them, and compounds that those compounds were around 20 million compounds. So after we used the filter of the pharmacophore, these 20 million compounds became 1 million, sorry, okay, here, 1 million, yes. So these million compounds share the pharmacophoric characteristics that could potentially give them insecticide activity. After this, we took the 1 million compounds and performed structure-based virtual screening using the GABA receptor. And these million compounds, of these 1 million compounds, around 3,000 of them showed high affinity for the binding pocket of isoxazolides. So of these nearly 3,000 compounds, we analyzed them. We did a human-based search of these compounds to see which one of them could have reported insecticide activity, but not known target, or that were potentially binding to GABA receptors. So we analyzed them, and we found that so we found among these compounds a member of a family named quinazolines that is a family that has several insecticides reported. Here I show you. I'm sorry, I don't know if it is me that is not seeing the presentation, or if it is delayed for everybody. Okay. Now we found a member of the quinazoline family that had high affinity for GABA receptor of Aedes. Now we did a bibliographic search of this family. We found several members of it that presented insecticidal and larvicidal activity, but they had no protein target reported. So they had activity, but they didn't know where these compounds bound for such activity. So we wonder if this quinazoline family could target GABA receptor. So to answer our question, we use the extractors obtained from the docking of these members of the quinazoline family, all of the 6 members. We dock those compounds in the isoxazoline binding pocket, and we embedded the complex of the receptor with the compound in POPC bilayers, doing the same protocol we perform with isoxazolines. And we did the same analysis. We analyzed the compound's stability at the binding site, calculating the root mean square desviation of these compounds in the plane of the receptor, and also in the axis, in the vertical axis of the system. And we found that they remain mostly tightly bound to the isoxazoline site. And we analyzed the hydrogen bonds that we had determined for isoxazolines, and we found that they were conserved in the interactions of quinazolines. And finally, we calculated the free energy binding of all quinazolines with the GABA receptor, and we found 3 of these compounds that have binding free energies in the range that the one observed for isoxazolines. Okay, so this gave us an idea that effectively it can be proposed that quinazolines target the same binding site as isoxazolines, and this indicates that quinazolines could be a good scaffold for developing new GABAergic insecticides that target that particular site. Now we are in the process of determining the larvicidal activity of the compound that we found in our virtual screening, that is the zeta. Sorry, I won't be telling the number correctly in English. So this compound activity we are measuring with Aedes aegyptis and trypanosomas to see if it has insecticidal activity. Okay, on the other hand, I told you that we were interested in searching for insecticides that are highly toxic for invertebrates, but that present low toxicity to mammals, and to analyze if quinazolines could be selective. We performed a new molecular docking, but this time we took the whole transmembrane region of the GABA receptor, not only the binding site where isoxazolines bind. And this is like a more relaxed docking, so it's called like a blind docking, where you explore all the surface of the protein, and it can tell you where the pocket is. Okay, but what we did is we did this docking with this very large box, and this allowed the docking to explore all the transmembrane region, and we performed docking assays of all quinazolines in the GABA receptor of Aedes aegyptis and in the GABA receptor of humans. this GABA receptor of human is crystallized, so we use a crystal structure that is in the protein data bank. And for the GABA receptor of Aedes, we use the same model that we were using in the molecular dynamic simulations that we saw before. And while in docking assays with GABA receptor of Aedes, all quinasolines bound to the isoxaline binding site. That is, they still bound to the same site, indicating that that is the site where they bound. Okay, so they don't bound anywhere else. They only bound to the isoxaline pocket. So this reinforced the suspicion that these compounds effectively are insecticides that bound to this site. And on the other hand, when we performed the docking assays with human GABA receptor, the quinasolines bound aleatory to all the surface of the receptor and never target the isoxaline pocket. That is, this would indicate that these quinasolines could be selective and could target insects but not vertebrates. So we conclude that this protocol that we applied of an hybrid ligand and structure-based virtual screening followed by MD simulations can be a promising tool for searching new GABAergic insecticides that target not only the isoxaline site, but we have been working with some other binding pockets. That the application of quinasolines for the development of pesticides have potential for the development of selective insecticides. Okay. This is the first part of what I wanted to show you. And now I'm going to continue talking about some non-specific regulations that could affect selectivity in insecticides. But if you have any questions of this first part, maybe you can, or any doubt that is short in this first part, we can do it now. I don't know how do you think, or do you prefer that we finish all the talk? And
yeah, I think it's better to follow, and then in the end we open for some questions.
Okay, perfect. Okay. Okay, now we were talking up to now, we have been discussing some interactions that are protein ligand specifics. So this is a protein recognizing, a ligand recognizing a protein that it targets, right, in its binding pocket. But as I mentioned before, we're working with a protein that is a transmembrane protein, and therefore the lipid bilayer plays a highly relevant role in this protein activity. We, in our lab, we study the modifications of the physical properties of the bilayers that can affect the partition of GABAergic ligands in general, not only insecticides but some others, allosteric modificators, and we have worked a lot with this characterization. But today I will not talk much about. I will not talk about that particularly, but I would rather show you some results. Sorry. I'm not seeing the presentation.
Yeah. Oh, no. Now I see.
Oh, okay.
Maybe it was too heavy in the archive.
Okay, there. So, as I mentioned before, the binding pocket of this insecticide, the isoxaline family, is located deep into the bilayer in the interface of the protein with the lipids. So it is highly probable that all compounds that target this site partition into the bilayer first, and then they access to its pocket. And this is important to take into account because of the differences that mammals membranes and insect membranes present. While mammals membranes have high concentrations of esterols, particularly cholesterol, insects cannot synthesize sterols, and they only incorporate them through the diet, so they have minimal concentrations of these sterols. And on the other hand, insects membranes have high concentrations of phosphatidyl ethanolamine, or POPE, and that is a phospholipid. And this makes clear differences in the compositions of both vertebrate and invertebrate membranes. So we wanted to know if these differences had any effect in the kinetics of access of insecticides to the binding site. So we performed molecular dynamic simulations that, like we did before with the receptor, that is all atom MD simulations. These simulations are all atoms are explicit in the simulations, and therefore they are highly detailed simulations. And we analyze the partition of these compounds into these two types of model membranes. We perform free diffusion MD, where and umbrella sampling simulations. Umbrella sampling simulations are simulations that allow you to determine the free energy of a path, chemical path. In this case, is if you have two atoms, the blue and the red atom, and you pull one atom away from the other, you can do molecular dynamic simulation of each one of these steps of the path, of each distance, and you can obtain the free energy of each of these steps, which gives you the idea of which is the minimal free energy of the system. Okay, so what we did is, in this case, instead of two atoms, what we have is the lipid bilayer and our insecticide, and we determine the free energy of the insecticide entering the bilayer. So this gives us the idea of where the minimum energy of the system is. So what we wanted to know is where the insecticide, in this case fluoroaranel, has its minimum binding free energy in the bilayer. And we did so with control membranes of phosphatidylcholine phospholipid that are the basic, like the control membrane. And we saw that this is the water and this is the lipid. These are the colors that we use to identify each part of the lipid bilayer. And we saw that the insecticides partition into the region of the glycerol of the phospholipid. On the other hand, when we use mother membranes with high concentrations of cholesterol, where we have 50% cholesterol in a PPC bilayer, the insecticides partition to the bilayer, but its minimum is located in the region of the phosphate group. Therefore, it stays in the zone of the charge region of the bilayer. And its partition to the hydrocarbonate change becomes desfavorable. On the other hand, the partition of the compound into a highly of a membrane with high concentrations of POPA, such as insects membranes, this partition is favored to deep into the bilayer in the region of the hydrocarbonated chain. This gives us the idea that the insecticide partitions different into vertebrates than in invertebrates bilayer. We also did 3D fusion molecular dynamic simulations where you have the bilayer and you have the insecticide in the aqueous phase, and you let them freely move and see where the compounds, where the insecticides partition, and we analyze where the compounds, where the fluoralanar molecules were partitioning. Okay, we did that with fluoralanar, and we saw that in in membranes with high contents of cholesterol, the fluoralanar molecules aggregated in the bilayer, while in POPC, POPE bilayers, the insecticide did not aggregate it and was solubilized in the lipid phase. Also, we saw that, why is this important? Because this gives us an idea that in cholesterol bilayers, this insecticide could has its access to the binding site could be inhibited because of its aggregation, giving some selectivity. So this could be diminish the toxicity of fluoralanar in vertebrates, because the insecticide, when partitions to the bilayer, it aggregates and is not capable of reaching its binding site, while in vertebrates it can do so. Okay, and on the other hand, when the compounds partition into the bilayer, we analyze the tilt or the angle of its disposition in the bilayer, and we could determine that the tilt that the insecticide has when in invertebrates membranes is the same as the one that has when it binds to the receptor, that is the dotted line. Here is the dotted line. We have the tilt of the fluoralanar bound to GABA receptor. So in POPC and POPC, POPE bilayers, it has the same tilt, while in in bilayers with high content of cholesterol, this tilt is different because it has angles that are around 20 degrees. That is the angle that the insecticide has. Also, is different in high in cholesterol containing bilayers, affecting its access to the receptor. Here is how it's the fluoralaner bound to the transmembrane domain of GABA receptor. We can conclude that the difference in the membrane composition could unspecifically influence the selectivity of GABAergic insecticides, modulating their solubility and access to the receptor, and that this non-specific regulation should be taken into account when we are evaluating the design of new insecticides. Okay, and that would be all.
Okay. I would like you, Dr. Virginia, for your very interesting and important investigation that you presented for us. As usual, we spend some time for some questions, for suggestions. Yes. If someone wants to ask something about the presentation, then we have still some time to do that. I had only a curiosity about it. As far as I understood, of course, your group team was trying to find some selective insecticides that would be less toxic for humans and more toxic for the insects.
Yes.
Then, do you have in mind, or I don't know if you are doing that, to collaborate with the synthesizing some kind of molecules that share some specific core that you have seen that's important for this selectivity, and to try to synthesize these compounds and then test them? I don't know if it's a kind of objective of the work.
Yes, we are very interested in matching the in vivo activities and the synthesis. That's the part we lack in our lab. We have the facilities to do biophysical assays to determine membrane interactions and the bioinformatic part of the design, but we do not have organic chemistries that do the synthesis, and we think that this quinazoline scaffold could be a good scaffold for developing new insecticides, and we're interested in validate them experimentally also. We have some collaborations with an organic chemistry that not for these compounds. He synthesized other compounds and they have measured these compounds insecticidal activity, and he does that. He synthesized other family of compounds not related to GABA, and they search for activity, antibacterial activity, insecticide activity, various tests. And he has some compounds that has found that are insecticides, and we're trying to see if GABA receptors are the target. But we do not have people that we can give our compounds to synthesize. Yes, that part we do not have.
Yeah, I understand. And I think that could work also for natural products or for
yes,
plant extracts or something like that.
Yes, we initially started working with natural products that act as that belong to the propofol family, that act like anesthetics, different types of activities that are also part of GABA's activity. But when we move to insecticides, we have not worked yet with natural compounds, but that would be the goal, to go for natural products. Yes, definitely. Yes.
Okay.
I know that maybe I don't know how much of the bioinformatic techniques we used were clear, or maybe I didn't go much deep into them, but I wanted to give a more global idea of our work. Maybe that was, I don't know if that got some of the ideas lost because of the techniques, but
okay. Any more questions? Well, I would like very much to thank you, Dr. Virginia, for your availability to share for us your knowledge, to make a seminar for us here in Brazil. And in the name of our post-graduation program, I thank you to be here and to do a speech for us
okay.
in our seminars. And good work.
Thank you for the invitation. And if anyone has any doubt that it arises after the finished talk, you can write me. I have no information. And here is my mail for anything. Okay. So.
Okay, that's important.
Okay. Well, thank you, everybody.
Bueno.
See you then.
Tchau, doutora.
Goodbye.
Nos vemos. Tchau, pessoal. Então, nos vemos, nos falamos.
Obrigado.